So last time we were talking about cells, and as I said, we're going to continue to talk about these cells, right, and these different types and their functionality. So remember the granulocytes, they all end in fill, they all have this characteristic multi-lobe nucleus, and it's really the different granules, basically vesicles containing different substances, is why they stain differently and why their functionality is a little bit different too, right? Basophils and eosinophils are kind of opposites to each other. Eosinophils are seen to increase because probably their substances inside their granules are effective against parasitic worms. Where you're going to see both basophils and eosinophils as far as allergic reactions. Basophils contribute to inflammation, eosinophils kind of quiet down inflammation. And then remember, neutrophils are really important because these are the ones that rush to the area during inflammation. They're called to that area. They're one of our main phagocytic cells. They're very good killers, right? They have many other things in their arsenal for eliminating. When we talk about our mononuclear phagocytes, monocytes is the name given to it when it's circulating in your blood. When it's in your tissues, it's referred to as macrophages. And so these guys are your first line of defense. They're there already in your tissues, your main phagocytic cells that are going to gobble stuff up when stuff gets past those barriers, our skin, our mucous membranes, right? The important thing for dendritic cells, they're there too, but they're more important to the next step and not sharing that information with the adaptive immune response, with those lymphocytes, the B cells and the T cells. And so they're sometimes referred to as the scouts, right? They're gathering information at the front and then they bring it back to the areas where the B cells and the T cells are located to share that information. So B cells and T cells we went over too, how they kind of are their names, why it's a T, why it's a B. And we're going to go over B cells and T cells very specifically when we get to adaptive, because they're the main ones responsible for that adaptive response, that response that changes over time. These guys are highly specific in the antigens that they recognize. Where natural killer cells, although they're lymphocytes, they lack that specificity. They need different components of our immune system to alert them to do that killing that they do so well. So remember, for these cells, the most important thing, right, is their functionality. What are they doing, right? How do they help us? So one of the ways in which they can actually do their jobs is the fact that they have surface receptors on their surface that help them interact with the substances, the pathogens, and the other cells in our immune system. And they communicate with each other, right? Cytokines are that chemical message. We've already kind of mentioned them. So they're released by cells of our immune system as well as regular cells. And they're going to communicate a certain message, right, to the receptors on cells that can actually do something about the situation. Adhesion molecules, right, movement of these cells to particular areas is very important. And so we've already seen adhesion molecules in action during inflammation, right? This allowed the neutrophils to grab on to the endothelial cells, the receptors on those endothelial cells, and actually travel into the tissue out of the bloodstream. So, we sense our environment, right? We use our eyes, our ears, our nose, right? We feel, we taste. So for them, it's a little bit different. It's all a chemical type world, right? They need receptors to be able to sense what's going on. They communicate differently, right? They communicate chemically with cytokines. They grab onto things not with hands, but with molecules, right? With receptors specifically designed to grabbing onto things, these adhesion molecules. So sometimes those receptors are equated to the eyes and the ears, right? How they see and hear their environment and sense it. How they communicate with each other, their voices are their cytokines. And their adhesion molecules are what are allowing them to grab onto things. So equivalent to how we use our hands a lot of the time. So surface receptors, these are receptors within the membranes. They're proteins, right, that are embedded in those membranes. Things are going to bind to them. And that particular binding sends a particular signal to the internal mechanisms of the cell, all the way to the nucleus, right, the command center that determines what's going to happen, when it's going to happen. So the name given to the molecules that actually bind to receptors are referred to as ligands, right? But just like as we talked about before, like in the endocrine system with hormones, right, that hormone, or in this case the cytokine, binds to a specific receptor. Right? And that's going to send a particular signal to that cell, and it's going to respond in a specific way. So this is general terminology, right? Ligands and receptors. Cytokines, there's a whole book on them. So we're not going to memorize necessarily individual cytokines. You guys would go cuckoo bananas, so would I. I don't even know them all. At one time I knew quite a few of them, though. So this is the general name, as I said, like hormone is the general name, right, for the chemical signals used by our endocrine system. Cytokines is the general chemical name to the chemicals that are used for signaling for the immune system. So some of them are based in classifications, based on their general functionality. So there's a whole class of cytokines that are chemokines. They're involved in chemotaxis, right? They're sending out the signal for particular cells, come here, right? And we know this happens during inflammation, right? There are particular signals that are sent out that tell the cells, come here, right? I need your help now. In our bone marrow, where all our red blood cells and all our white blood cells are made, there are stem cells that can generate any one of those cells. Well, how the stem cells know what cells we need at any particular time is due to colony stimulating factors. So this is a whole set of cytokines that specifically tell the stem cells we need lymphocytes right now, or specifically we need B lymphocytes, or we need neutrophils, or we need macrophages. Well, that's going to go down that lineage and produce that subset of white blood cells. So that's why it's referring to colony, right? So it's a group of cells that are similar. So remember, leukocyte is just another name for a white blood cell. Interferons are very important for viral infections. So this is the signaling that our cells use when we have a viral infection. And so you'll notice too that we've utilized this. We actually purposely give people additional interferon sometimes to help treat certain viral infections, right? And there's other treatments, like if you listen to the commercials and stuff on TV, right? Some viral infections, and I think the one that I keep seeing over and over is a new treatment for hepatitis C, right? So this is a viral infection, and it says that this particular treatment doesn't include interferon, right? So it's dealing with the virus in a different way. How, I do not know. Of course, they don't tell you that on the commercial, right? That's a little bit more information than most people care to necessarily know. Interleukins, there are a lot of these, and so they're commonly abbreviated IL. So the most common one you hear is interleukin 2. They're numbered, right? IL-1 and IL-4. This all stands for interleukins. And again, leukocytes are white blood cells. So this is specifically communication between our white blood cells, talking to each other. And this class, as I said, there's lots of them, lots of numbers, lots of interactions that we're still studying. And these chemical signals, this communication is very important to the functionality of our adaptive immune response, right? Those lymphocytes talking to each other, the macrophages, the dendritic cells sharing the information they've gathered at the front lines. Tumor necrosis factor, as the name implies, this is a chemical signaling specifically for killing tumor cells. And so in this case, when our cells are behaving badly, right, we're going to actually elicit inflammation we're going to call up the army, we're going to go in, and we're going to try and eliminate these cells that aren't doing what they're supposed to. So definitely we want an understanding of these general classes. These are very important, right? But we're not going to memorize individual, like they have listed here on this table. We're not going to memorize individual cytokines, like, for instance, as I said, IL-1, right? We're not memorizing that it comes from macrophages and epithelial cells, and it's going to activate macrophages and T cells and induce fever, right? We're not going to go to that level. We're going to stay at this level, right? General categories. But as I said, notice that this is communication, right, with white blood cells, and a lot of communication with T cells and B cells listed. No, that's what I'm saying. You don't need to know these, right? I don't want you memorizing these. But understand the interleukins in general, right, is communication between white blood cells, and really definitely the ones part of our adaptive immune response, those B cells and the T cells, as you can see by these specific examples given here. And as you can see, interferons, there's more than just one type of interferon. And again, it has to do with who's communicating this message. So adhesion molecules, as I said, we've already seen these in action, right? These allow the cells to literally adhere, attach. These are receptors on their surface. This is so those phagocytic cells, neutrophils, more macrophages can head into that area when we have inflammation. That's when these receptors are expressed on the endothelial cells, and the adhesion molecules that are present on our white blood cells are going to grab onto these and use that to help facilitate their movement out of the bloodstream into the tissue. And so we watched that video where you literally saw them rolling along as they were interacting with the endothelial layer and then squeezing through. So how do they sense their environment? They got to sense that environment by receptors. So there's a whole bunch of different types of receptors and receptor systems that our body has for detecting bad stuff. Toll-like receptors, right, are receptors that are specific for danger signals, right? I mentioned this briefly before. The complement system is a series of proteins that help us recognize when invasion has happened. And this one can actually cause particular things to happen. So once it's activated, and there's different ways it can be activated, what are the outcomes? What happens? What happens when we detect long double-stranded RNA, right? We don't have double-stranded RNA in our cells, do we? Now what type of invader might be double-stranded RNA? Viruses, right? That's one of our specific detections for viruses. So we have systems within our blood, right, that can detect signs of tissue damage or microbial invasion. They respond to patterns associated with danger, right? And they can some of them can directly destroy the invader, or they're going to recruit other cells, right, other host defenses to help eliminate this particular invader. So lots of acronyms, right? The important thing here, right, is being able to detect these foreign substances, these danger molecules that could harm us. So the important ones here, right, so these are receptors that recognize general patterns, right? So they're abbreviated PRRs, for pattern recognition receptors. They can detect pathogen-associated molecular patterns. Well, what would that be? So this is the important part right here. Oops. It's not, I wanted to highlight it. Oops, that's the wrong thing. That's not a highlighter. Where's my highlighter? I have no idea. Oh well, no worries. Right. No highlighter. I don't know why. Used to have a highlighter. Ah, here we go. I found it. I found it. Oh, it's a different kind of highlighter. That's crazy. Alright, we'll undo that. Alright, that's more than I can handle. That's freestyle. Okay, so what are these things? These are cell wall components. Think about that, right? For bacteria, they have certain components. We learned about lipopolysaccharide is part of that outer membrane for gram-positive or gram-negative. Gram-negative. Peptidoglycan and lipoteichoic acid are probably the outermost covering for gram-positives, right? Those are components associated with them. Lipoproteins, flagellin subunits, right? The particular protein that makes up the flagella of bacteria. Viral RNA molecules. These are all things that are definitely not us, right? This is definitely stuff that's foreign. This is definitely stuff that could potentially harm us. But these are general patterns, right? This doesn't tell us that this is E. coli or salmonella, right? It just tells the body that this is a bacteria, right? And probably a bad guy. It's not specific, it's general. I'm going to get my pointer back. There we go. So in this video, right, they go over some examples of toll-like receptors, right? And when you look at it, again, notice that list. But one thing I want to point out to you guys is, see how there are some on the surface? There's one that's inside. Which one is that one? It's toll-like receptor number 3. So when you look over here, that's the one detecting viral double-stranded RNA. That being on the surface would do us no good, right? So we even have receptors, although we talk about ones on the surface, we even have ones inside of our cells, like this one's associated with an endosome. Others will be within the cytoplasm. And of course this one needs to be inside of our cell because that's when it's going to come in contact with the RNA from the virus. The virus, all viruses are at least surrounded by a protein coat. They're not going to decode until they're inside of our cells. So that's when our receptors would be able to come in contact with that viral double-stranded RNA and be able to detect and alert our cells that an invasion has happened by a virus, specifically an RNA virus in this case. So what's going to happen, right? So we have these receptors on the surface, inside the cell, inside the cytoplasm. They're going to induce gene expression. This information is going to be carried all the way to the nucleus, right? To our DNA, to our command center. And it's going to respond accordingly. Of course, the most common response, right, when we have a danger signal is inflammation. Get more white blood cells to the area so we can eliminate the invader. If it's viral, though, there's a specific response against viruses. Right? It's a little bit different than inflammation. Make sense? Right? So the alarm goes off, we call the infiltree in. Right? Come help us. But those receptors are going to be our detectors. They're going to send off the alarm. Right? So NOD-like receptors, again, just a set of receptors. In this case, they're inside the cytoplasm, inside the cell. So we have multiple layers of protection, not only on the outside of our cells, but on the inside of our cells to detect when stuff gets in. Because some bacteria are intercellular parasites. So we want to be able to detect, like we said before, peptidoglycan, right? Flagellin. Even single-stranded RNA that can be associated with particular viruses. We process our RNA, right, in a way that our cells recognize it. So if it's uncapped, which means it's not processed, then we have receptors that detect that and would let us know that this is probably a viral infection, as opposed to our own single-stranded messenger RNA. So complement system is another system of detection. Right? And so I had asked you guys to watch these ahead of time, right? So you'd have some familiarity with it, that I probably wouldn't have time to play them in class, and I certainly don't. But they're there for you guys. So the complement system is a series of 9 proteins, and so C designates complement, and then they're numbered. They're unfortunately numbered in the order in which they were discovered, not exactly the order in which they're necessarily activated. Some of them split into pieces, and so the fragments are labeled A and B. So for instance, C3, that particular protein in the complement cascade, splits into C3A and C3B. And this always happens, right? We're always producing C3A and C3B. When C3B binds to the surface of, say, a bacterial cell, this initiates what we call the alternative pathway of initiation for complement. Remember I may have said before B for binding, right? So I always remember C3B, B for binding. It does what we call opsonization, right? We mentioned this previously. When they're coated with this C3B, your phagocytic cells, right, your neutrophils, your macrophages, your dendritic cells can grab onto C3B so that they can then engulf that bacteria into a phagosome, fuse its lysosomes with it to form that phagolysosome and destroy that invader, right? The first part of phagocytosis is actually being able to recognize and attach to the invader. So C3b, this component of this complement system, is really paramount in helping out with that recognition and attachment that needs to happen. And so if we have C3b that's there and it binds to the surface of the cell, and so any resident macrophages that are there on scene, they're going to engulf it. But we don't want to just stop there, do we? Depending on how many got in, right, you may need to call in the infantry. You may need some more macrophages and definitely some neutrophils to get rid of the invader. And so some of the other components, like the C3A that's not binding, help elicit inflammation. They're going to call the cells to the area. And then, as I said, C3b specifically is an opsonate. It will actually bind, tag those cells for destruction. One of the other outcomes of this system is actually producing a complex that will lyse the cells. Right? So this is what can happen when complements become activated, right? When it's been triggered. We'll get inflammation, we'll get influx of more cells to the area. If C3b is binding, it's going to opsonize, it's going to tag those cells for destruction. And then we could also form the complex that will destroy the cells. So C3a and C5a, right, which are produced in the activation of this cascade, what happens is once one thing happens, the next thing will happen. Once this system becomes activated, it's going to go through the whole system. So it's like a set of dominoes, right? Once you get the thing started, it's just going to keep going till the end. So one of the products of that cascade is C3A and C5A. Both of these, right, are really strong inflammatory molecules. They're going to help assist in endothelial changes so that the cells can head to that area. And C5A specifically is going to call neutrophils, right? They're saying, come here, come here, come here. We need you guys.
So we need to learn these complements.
Yeah, just a few of these do you need to memorize, right? So C3A and C5A, it's important to know that those are the key complement proteins for inflammation. And C3b is the opsonizer, right? It's tagging for destruction. So as you see in this picture, as we've seen before, right? Those little orange squares represent C3b binding to the surface of the bacteria. And then notice there are receptors specifically for C3b on our phagocytic cells, on our macrophages, our neutrophils, our dendritic cells. So they can grab, they can recognize, and they can attach. These letters you don't need to memorize, and numbers. But know the name that we give to this complex. It's called membrane attack complex. It's commonly abbreviated MAC. Not a Big Mac from McDonald's, right? But membrane attack complex. And this is important because it inserts into what? Membranes, right? So it'll insert into that membrane, it'll create a pore and basically cause that cell to lyse, that foreign cell. So as far as bacteria are concerned, this is more effective against gram-negative and a little bit less effective against gram-positive. Why is that? What do we know about the membranes as it relates to gram-positive and gram-negative? Gram-negative have that outer membrane, right? And it is a membrane, right? So this complement, this membrane attack complex can attack that outer membrane. Where for gram-positive cells, it'd have to travel through what to get to the cell membrane? Through the cell wall, through that peptidoglycan layer, right? Not to say it can't be done, it's harder though, right? And so your gram-negatives are very susceptible to this MAC complex, this membrane attack complex. And again, once the process is initiated, these proteins insert into the membrane and create this donut-shaped structure, this membrane attack complex. And so this is what it, similarly to what it looks like, right? Again, it's creating a pore. So all these little black dots are actually holes in the surface of that cell, right? Where the MAC complexes have inserted into that membrane. Now this happens to foreign cells. So this happens when you get a blood transfusion too of the wrong red blood cells, right? Your immune system will recognize it as being foreign and it'll insert MAC complexes into them and destroy them. The bad news is the reason why you get a blood transfusion is because you need those red blood cells, right? Not that you need them destroyed, right? But again, foreign things, things that are not you, your immune system is going to attack. And this is one of the mechanisms it has at its disposal. And this is part of your innate response, right? So you're born with this. You can do this from day one. If you're talking about plastic surgery, like tissue grafts and things like that, yeah, it's foreign, right? So your body responds to it as if it could be something that'll hurt you. Yeah. So she was saying if you have plastic surgery, so like if they're putting foreign stuff in you, like breast implants and things like that, right? There could be a chance that your body recognizes it being foreign and attacking it. So, yeah, it's like I said, it's not worth it, y'all. You're perfect the way you are right now. You don't need to change you.
I had a friend of mine, she did the butt injection thing.
Oh, God.
Yeah, and she, I think her body got infected and she had to go to the hospital, and I mean, like, they had to literally chop off, like, oh, it was awful.
Yeah.
But she had to go through to get it out, and just, like, so I started kind of purple and swelling up.
Yeah. Yeah, like I said, it's not worth it.
They had to tell her they were going to have emergency surgery, they like literally cut like tissue out of her body.
So this system, as I said, are proteins circulating in your blood and your tissue fluids, right? So it's mostly in the inactive form, right? It's got to be activated for this system to move forward, right? For the different proteins to break into different pieces and to do the different jobs. They can interact with components of our adaptive immune response. So one of the most common products of our adaptive immune response is antibodies, right? And so antibodies can also interact with this system, right? And that's one of the ways this system can actually be activated. So that's what they mean by augment activities of the adaptive immune response. Stimulation of inactive proteins initiates a cascade of reactions. Like I said, Once it starts, it just keeps going, right? It's like a domino effect. So how do we get this system started? There's 3 different pathways and that we can get the system started. We can activate it. There's the alternative pathway, which again, these names have to do with when these things were discovered, right? So for instance, the classical pathway was probably the first way that this thing was initiated. But in your body, it's probably being initiated more often at the beginning of infection via the alternative pathway. So alternative sounds like it's something that we do alternatively, right? But it's probably our primary response. The classical is just what we discovered first. And then you have the lictin pathway, which is a very specialized pathway specifically for recognition of lictin. So the lictin pathway is activated by special receptors that are special molecules that can be recognized by this system. So mannan-binding lictins, these are special proteins within our complement system. Again, it's a pattern we're recognizing. Mannan is a polymer of a sugar known as mannose. And it's very common in microbial cells. So again, not something we have, it's something foreign. And when the mannan-binding lictins bind to those microbial substances, this activates complement. And one of the main triggers in complement, right, that always happens is this C3 breaking into C3A and C3B. And then as we said, right, C3A is important for the inflammatory response, and C3B is important for opsonization, binding to the surface of those cells so that we recognize. The cascade will continue down. One of the major byproducts is C5A, which also is going to help in this inflammatory response. It's going to call the neutrophils to the area. We're skipping a lot of steps in here, okay? And then the other thing that can happen is the formation of that MAC complex, right, the membrane attack complex. So if they're foreign cells with an exposed membrane, it's going to assert in and lyse them. The alternative pathway is activated by C3b binding, right? C3 is always getting broken into C3a and C3b. C3b can attach to bacterial cell walls, right, and bacterial surfaces. When that happens, that's going to initiate the continuing of this cascade. So we get C3a and C5a, we get that inflammation. We get even more C3b binding for opsonizing, tagging these guys for destruction, and the potential of the MAC complex formations. That's the alternative pathway. The classical pathway is when antibodies bind to the surface of microbial invaders. There are components of complement that will bind to these antibodies and activate the complement cascade. So we get the C3a and the C3b, we get that inflammation response. We'll get C3b that will also help tag for destruction. Because that's what antibodies do too. They're very strong opsonizers. They tag them for destructions. Our phagocytic cells have receptors specifically for our antibodies. So when something's coated in an antibody, your phagocytic cells are going to go after it, grab onto it, and destroy it. And then depending, right, we're going to get some formation of the MAC complex too. Can insert into those cells' membranes. Now, antibodies are a product of our adaptive immune response. And so this takes time to develop. So that's probably why, for the most part, if it's an invader we've never seen before, we're actually probably having activation either the lictin pathway or the alternative pathway. Probably not the classical. Right? But classical does happen as well. And that's the main reason, too, why, we'll recognize foreign cells when you get a blood transfusion of the wrong type. It's those antibodies binding and activating complement. That's going to be one of the ways that those cells are destroyed. So why doesn't this happen to our cells? Why doesn't C3b bind to our cells, and why don't we activate complement. Well, obviously we wouldn't want to do that, right? We don't want inflammation, right, in response to our own cells. We don't want opsonization, tagging for destruction of our own cells. We don't want to lyse our own membranes with the MAC complex. So there are molecules in our host cell membranes. They're called binding regulatory proteins. They inactivate C3b. So if C3b binds, they inactivate it. They kind of keep it from being able to interact with the other complement proteins so that it can't activate the system. Unlike for microbial substances, they don't have these regulatory proteins, right? So they have C3 binding to them, and it activates the complement cascade via the alternative method. So guess what? Are there microbes out there that have found a way around this system? Of course, right? Always. Some of them are even can coat themselves in our regulatory proteins to stop C3b from binding. Right? Evil little guys. I might just have enough time for interferon activation. So remember, interferon interferes with viruses specifically. And this is, again, a cytokine, right? This is a communication between our cells, specifically when we have viral infection. So, again, within our cytoplasm, we have receptors, right, that are able to detect. And, again, we modify our RNA, right? So, even if it's not double-stranded, if it's single-stranded, we can detect. We have receptors that can detect that it's not our RNA. Right? And especially if it's double-stranded, it's not ours. So once a cell detects that it's been invaded by a virus, by detecting that foreign nucleic acid, those receptors, of course, are going to send that signal to the nucleus, and that cell is going to start producing interferon. Interferon is going to be released by that cell and bind to receptors on neighboring cells. And it's basically a danger signal, right? Like a fire alarm going off. If a fire alarm went off, you would not stay in the building, right? And the case for these guys, they get prepared. Cells that get that signal of interferon from a neighboring cell, they know, uh-oh, a bad guy has gotten in. Be prepared, right? It's like an alert signal being sent out on campus, right? They know there's a bad guy in the vicinity and they need to get prepared to fight him. And so they're going to synthesize specialized enzymes specifically to help them destroy a virus if it gets inside of them. Right? So they get ready for the potential of an attack. The bad news here is that when the virus, and if it does get into their cells, if they've activated this protection mechanism, they're going to destroy the virus, but they're also going to destroy themselves in the process. And definitely the one that was infected, it's going to get destroyed by the virus infection. Okay? But in the process, he's going to warn all of his friends nearby by producing that interferon, get ready. Right? The bad guys are here. You need to defend yourself. So we lose some cells to viral infection. But the good news is they are able to alert neighboring cells, and those neighboring cells can hopefully stop the reproduction process, right? If that cell in the process, of course, it dies, but still, it stopped the virus from multiplying. So therefore, it's going to stop the spread to other cells, right? Which is what happens with viral infection, right? They use one cell as a factory, they bust out of that cell, they go infect other cells, use it as a factory. You know, all along our cells are dying, right? If some of them die in the process of eliminating the invader, right, that's better than it just keep going. This process just keep going and keep going. So basically, some of them commit suicide, right, and to stop this continued cycle of viral replication. So again, notice, as I said, the cells go through apoptosis, programmed cell death, right? If the virus gets in, they're going to die, right? They're going to try and stop that cell for becoming a virus-making factory. Stop that virus from replicating so it can't infect other cells. So remember, apoptosis is programmed cell death without eliciting inflammation. So we're not going to call in neutrophils and stuff in this case. The cells are going to take care of themselves. They don't need backup. Hopefully they don't need backup. Are there ones that can shut down this system? Yeah, of course, right? We still have viral infection. I know my watch has been not keeping time well. I probably need a new. Okay, no, we're good. Okay, so this is the last topic for this lecture, so I want to finish it up. And maybe I'll post that other innate one for you guys, so that you can work on both of those connects depending on how your weekend goes, right? You know, the way the Saints have been going, just don't even bother watching, right? Do your homework instead. All right, so what are the outcomes of fever, right? So why do we do this? Why is this beneficial, right? Believe it or not, it actually is meant to be beneficial, right? So what fever is an increase in your core body temperature. And it's one of the strongest indicators of bacterial infection. And the reason for this is the unfortunate thing that some microbes produce substances, right, exogenic, so they're producing it, our body's not producing it. Exo means outside, right? Cryogens is the general name given to the chemical substances that induce fever, cause us to raise our core body temperature. So we produce our own cytokines, right, endogenous cryogens, to increase our core body temperature. But the bad news is some pathogens, specifically bacteria, also produce this substance. So we're heating up, they're producing this substance, and we keep heating up, right? This signal gets overexpressed, you could say. And so when you have a constant fever that's not going away, that's continually to increase, it's probably a bacterial infection, right? Definitely need to go to the doctor and probably get some antibiotics. Where if you have intermediate fever, it might be viral infection, right? Because sometimes even with viral infections, we get fever, right? And again, somehow our internal system is getting turned on to increase temperature. What is increased temperature? How does this benefit us? Well, if we get too warm, remember, how do we kill pathogens in our food? Most oftenly, what do? We cook it, right? And that's why we have certain temperature ranges for certain foods, right? Because we know potentially what microbes might be there and how hot we got to get to destroy them. And when you heat them up, you're going to distort membranes. You can coagulate, purposely destroy proteins, right? Basically shut down and disrupt those cells. The bad news with that, especially if we're not controlling the system, if a bacteria is causing us to heat up and keep heating up, is that we can get too hot for even ourselves, right? And we can destroy our own membranes and our own proteins. That's why fever can be scary when it goes too high and goes on too long. But it is a normal immunological response as fever. The also benefit, other than that we could get potentially too hot for the microbes, is that chemical systems, right? Chemical reactions, that's what our body is made of, that's what happens in our cells, happen faster when it's warmer, right? So we speed up, we get faster at our defense by being warmer, right? That's the benefit of fever, right? We're going to go faster, right? And we could potentially kill the microorganisms by getting too hot for them. So that's it for innate. Oh, that's what it